Pharmaceutical Processing, in conjunction with INTERPHEX 2016, presents INTERPHEX Live.
Good afternoon. All right, this is the afternoon session, day 2, INTERPHEX Live. So, what we did this year, this is actually a repeat from where we were last year. We're going to talk about what happens a year later. We had such an incredible amount of interest in this area, needless to say. So, but because I'm a little, well, comes from being partly Italian, but I keep forgetting things. But I think what we'll do is we'll start with what is a biosimilar, Parrish? We'll go from there. What is it?
What is a biosimilar?
What is it?
Well, as you know, there's been a lot of debate about what constitutes a biosimilar with regard to its similarity to the innovator product. And the definition of that similarity and the quality attributes of the critical quality attributes that define biosimilarity and comparability to the innovator drug has been a subject of hot debate in many regions of the industry and different territories, actually. So there is no one easy answer to that question. And as you know, biocomparability or comparability of a biosimilar to the innovator drug is proven at different levels, right? There's first the analytical level, and then there's potentially, if differences are seen, there's potentially in vivo evaluation of the comparability to the innovator product, and then ultimately there could be even human clinical validation to verify the comparability of the biosimilar to the innovator product. So there are levels of comparability, and within those levels, there are actually different definitions, fine points across different organizations and regulatory agencies.
Okay, well, that sets the stage, right? So here we are a year later. So Manny, how much progress have we made here?
I think we've made measurable progress with regards to biosimilars. We've certainly had approval of a monoclonal antibody in the US recently. That's measurable progress. We've had etanercept approved in Europe. That's measurable progress. So I believe it's not a massive quantum leap, but certainly we're making measured progress in this area. We also have a lot of innovative companies jumping into this fray of biosimilars. So all of those things point to us moving in the direction of getting more and more biosimilars into our pipeline and into customers' hands.
I see. I was remiss, sorry. Manny Krishnan is with Pall Corporation, right? Parrish Galliher is with GE Healthcare in Upstream, CTO of Upstream. That's my screw up entirely. Sorry, gentlemen. I forgot I should have done that first, but I really wanted to get moving on that. So let's get back to the topic at hand, and that is, so Parrish, so obviously some progress has been made. What is difficult here? How difficult is it for somebody to say, this is what I want as a biosimilar? How big is that hurdle?
Well, that's the $64,000 question. The folks that are getting into biosimilars who have been producing the innovator molecules, of course, are most informed about what's involved and what it takes. Those who are at the other extreme, who have never been involved in biologics before, and there are many examples of that. For instance, small molecule companies that are interested in getting into biologics have to create the entire knowledge base regarding the critical quality attributes, of the molecule, the process science technology to produce that molecule, and then the validation work to prove that it's biocomparable to the innovator molecule. So, and that's a lot of infrastructure and a lot of process science and analytical knowledge that the new company has to bring to bear on the project. And that's a very large investment, and that does not happen overnight. And there are a lot of subtleties that can lead to surprises along the way.
Well, I think what I wanted to do then, so let's look at it. Here's what I think we'll do. So we'll look at, so Parrish, let's look at the difference between developing a biosimilar and starting with a new compound, and then we'll get into the challenges that are associated. This way we can, Manny will handle that. So let's talk about, how difficult it is to what's the comparison between a biosimilar and a when I'm going to look at it, what's the differences in the high
Right. So in a very simple way, we can sort of make an analogy to if you're developing a biosimilar, you're essentially trying to repeat somebody else's football game and kick your football through the same goalpost a number of times. Goalpost defining the quality attributes, the clinical efficacy and safety, in vivo, PK/PD, et cetera. And so that's sort of the very simple analogy I like to use from my own common sense about what it takes to develop a biosimilar. You're basically recreating what somebody else has already done and trying to kick your football through the same goalpost. If you're developing a new drug, obviously there are no goalposts other than what you have established as the critical quality attributes, as what you've established for efficacy and safety in concert with your regulatory agency, and it's all new territory. So you're not trying to repeat or reproduce what somebody else has done. You're basically carving whole new ground with your new molecule. So you have a certain amount of freedom, additional freedom, in the development of a new molecule versus a biosimilar.
So Manny, it sounds to me like, you got some constraints there. So how do we, what's the challenges like?
So, so you have constraints from a technology standpoint in terms of, as Parrish very eloquently put it, hitting the goal between the same goalposts. But the additional constraints as well with biosimilars, which are probably a little more profound. One of them is, at least at the current point in time, not quite understanding what the market acceptability is and therefore what the market demand is for the biosimilar. So how do you set up a manufacturing process? How do you set up a manufacturing facility with these unknowns? In addition to the unknowns that Parrish talked about in the development process of it, how do you go to market with it? How do you develop a process for it that meets the market requirements, market expectations that are largely at this point in time not very well defined? So those are additional challenges that come with biosimilars as well.
So what are we doing to hit those challenges?
So a lot of the technologies that have been implemented in traditional molecules are actually finding place in the biosimilar world as well. You see a lot of companies, particularly in India and China, they are setting up their biosimilar facilities using largely single-use technologies. What single-use technologies do is provide them a significant amount of flexibility to gear up and gear down, depending on what the market demands are. So there are other technologies also that companies are developing that actually play very well into this biosimilar world because of this unknown about the market demands.
So Parrish, maybe I ask you a question then. So obviously the biosimilars, they seem to be having an impact on biotechnology, period, biomanufacturing technology, right? I mean, what's your take on that?
Yeah, I think we're continuing to see that the biosimilar era, as it were, is going to push the boundaries of manufacturing efficiency with regard to cost of goods, speed to market, and then flexibility in facilities that are making these biosimilars such that they can switch from one biosimilar to another. Because many of these facilities that we are building and others are building around the world are providing drugs or biosimilars to local territories whose quotas are not large enough to occupy a whole facility for an entire year. So as a result, in order to maximize the utilization of these facilities, they need to switch to other molecules throughout the year to minimize their overhead costs and amortize all of their fixed costs. We think that biosimilars are going to continue to drive optimization, efficiency, innovation in bioprocessing, and utilization efficiency improvements across the entire business.
So Manny, that's how you see this?
Absolutely, absolutely. I mean, I think there's been talk in the industry about continuous processing. Continuous processing, we've recently heard about the FDA approving Pfizer's process and Janssen's process for a continuous processing of a small molecule. However, we also see this moving into the biological world as well. We see continuous processing as addressing some of the things that Parrish talked about, effective utilization of manufacturing capacity, giving the flexibility that the users need in order to flex up and flex down their manufacturing processes, and also remove waste from the manufacturing processes.
Yep.
So all of these things, reducing, increasing their return on invested capital, improving their flexibility, reducing the overall cost of goods, all these things come from looking at new ways of processing and not using the traditional manufacturing processes that have been tried over the last 20, 30 years.
Right, and I think between small molecules and biologics, you got to remember when we talk about continuous process, that's really for you guys the fill finish part, you know, because basically we're granulating, densifying, compacting, and coating.
Yep.
That's pretty much what Janssen's
Yep.
continuous process is.
Yep.
Presently, we don't even reach that far back other than
Yep.
maybe milling or screening a drug substance. Having said that, I think you guys got some unique problems. So, you know, I mean, so what skills are you looking for there, Parrish? You know, obviously with the biosimilars, and I mean, what skill sets do you put in place over at GE? I mean,
Number one has to be analytical characterization capability. And it can't just be the release assays that you would do to release your bulk drug or your drug product. We're talking about characterization of the molecule at the microheterogeneity level, whether it be glycosylation, a secondary structure, tertiary structure, quaternary structure, and then obviously in vivo characterization for PK/PD in animals, if necessary. So analytical capability is clearly number one. Number two would be the ability to develop a process that produces the molecule that, again, lands between the goalposts established by the innovator company for that particular drug. And doing that potentially with new technologies, be they single use or continuous manufacturing that are running more efficiently, and maybe not even with the same cell type or cell line, and still hitting the goalpost for all the quality attributes that are critical for clinical efficacy and critical and relevant to clinical efficacy and clinical safety, which brings up a whole nother point, which is the ability to distinguish between what differences in the molecule are important clinically or not, and safety or not. Those are the ones that matter. So the ability for medically to distinguish those factors is another critical skill needed in these companies.
So obviously it's a team. So what else? So you must a significant look at clinicians and
analytical experts, process experts, medical experts, obviously operational experts for operational efficiency, process technologists for new automation, new innovation in manufacturing, low-cost manufacturing, et cetera.
Okay, so Manny, let me ask you one here. I mean, you know, if I got to hit it through the goalpost, obviously I got to be playing with the same shaped ball on the same field. So how critical is it that I'm using the same process for a biosimilar as an innovator, or do I start, I mean, you know, we got this constraint, but are we constrained all the way through? I mean, what's your take on that?
I don't think the regulators are insisting that the same process be used. In fact, for some of the simpler biological molecules, the regulators are even going as far as saying if you can show analytical comparability, that's actually sufficient. You don't even need to show clinical comparability. Right? For molecules such as monoclonal antibodies, that may not necessarily work out because you have an immunogenic response. But for simpler molecules, you can demonstrate analytical comparability, and that may actually get you to the goalpost. Right? So there's really not a need for you to demonstrate the process is the same as the innovator molecule, as long as you can demonstrate that clinical comparability exists and the analytical comparability exists.
Okay, so, you know, one of the things I always find interesting, you know, and again, not coming from my world, I look at the process, I look at the stuff I make to use, you know, is that all the same? But so from the biologics part, Parrish, I mean, this really meant, so am I limited in the cell line that I gotta use? I mean, seems to me you guys got more burdens than we got.
Well, the problem that biosimilar developers face is that the innovator processes are 10 or 20, 30 years old. And in many of those processes, you know, my team's actually developed a number of these, you know, in my career in the drug industry. And those processes were low titer. They were, less than a gram per liter in many cases, or 1 or 2 grams per liter. Today, to be cost competitive, you need to be in the 3 to 5 gram per liter range. So how are you going to get there with the old process, with the way it was done? And this is the conundrum that many folks are facing. They're faced with the pressure to reduce cost of goods, to reduce pricing, to increase productivity, et cetera, in the face of changing the process from what it used to be, because those old processes are not as productive and cost competitive today. So that's the rub. And that's what really stresses the organization's skill sets, analytics, process technology, and medical relevance of the differences between the biosimilar and the innovator product.
So the per liter aspect, I mean, we had a couple of speakers in that talked about intensification. I mean, this is the hot topic now. Mm-hmm. Maybe we want to talk about that just a little more, Manny. Where do you see, I mean, if I'm trying to make something the same, words like intensification from my world make my blood run cold. So where are we at with that?
I think, like Parrish talked about, you have the pressures of trying to reduce your cost of goods. And you can't do that while maintaining the same process. So you do have to, you're forced to, in a way, innovate to make sure that you can make the molecule that can hit the goalpost while hitting your cost targets as well. So I think, to me, when I talk to a lot of the companies involved in the biosimilar exercise, they're less concerned about mimicking the process. They're more concerned about meeting the goal, getting the ball through the goalpost. So that's what their concern is, not necessarily keeping the process exactly the same. So I think we are kind of mature from that standpoint.
Okay, so
if I could just add to that,
yeah, please.
There's another distinction to be made here, and that is the difference between biosimilarity and interchangeability, which is a higher level. Interchangeability is a higher level of comparability to the innovative molecule. So the additional skill set is, again, distinguishing for the pharmacist who's prescribing, who's dosing the patient, and the physician who's picking between the innovator molecule and the biosimilar, whether or not that molecule, the new biosimilar, is in fact simply interchangeable with the innovator product, or whether or not it's not interchangeable, it's a biosimilar, and therefore may not be as efficacious and as safe and as desirable for the patient. In the latter case, that would mean that the marketing capability would be another critical skill set of a biosimilar company. If your biosimilar does not achieve interchangeability with the innovator molecule, then you're going to have to sell the physician and the pharmacist on the benefits of your drug over the innovator drug, because it is not interchangeable. So the holy grail here is to achieve interchangeability such that it is essentially blind to the patient. Doesn't matter. The pharmacist can switch from one to the other, and there's no debate about the value of one to the other.
Yeah, like for sure.
Like for like.
So
interchangeability.
Interchangeable.
Yeah.
So the hard sell is, well, mine's almost. Or, yeah, you could if you up mine by 1.5 times. I mean, what are we talking about? You got a higher dose. I mean, stuff that, all red flags, right?
Well, there are so many biosimilars, I wouldn't want to comment on anything specifically.
I'm not trying to put you on the spot, right?
But, I think that it's again, coming back to the goalposts. So, and this is where it gets expensive. You got to go and find enough lots of the innovative molecule and analyze enough of those lots to establish how wide those goalposts are for as many of those lots as you can get. And obviously, the more lots that you can analyze, the wider those posts become, and the easier it is for you to kick your ball through those goalposts for each one of those attributes. And there are many quality attributes. Each one of them has their goalposts, and they're all different widths. So, this is the challenge. And every molecule is different. The CQAs, critical quality attributes, are different for all the different molecules. There are simpler ones like filgrastim, GCSF, and there are very complicated ones, highly glycosylated, like EPO, where the glycosylation is critical. And ADCC activity mediated monoclonals, also glycosylation, very complicated and critical to efficacy and clearance rates and PK/PD. So there's a huge range of complexity across these issues, depending on the molecule of choice.
So Okay, go ahead, Manny.
I mean, I think the challenges also exist in small molecule world, right? I mean, if you look at sustained release or extended release formulations, they have the same kind of challenges in terms of demonstrating equivalence of a generic drug to the innovator drug. Mm-hmm. So, I mean, to me, these are challenges that exist but can be overcome.
So that's a good point. A formulation is another way to differentiate.
Yeah.
So some drugs maybe should be sub-Q instead of IV. Or can you dose them at home? Would it be easier for the patient to be able to dose themselves, you know, et cetera, et cetera. So that's another avenue that marketing folks are researching to differentiate. If they don't achieve interchangeability, how can they differentiate for a biosimilar?
Yeah. Well, let me, you know, one of the things that, you know, we, I gotta tell you, coming from the small molecule world, one of the things I shamelessly plagiarize and quote all the time is how we should scale out like all our buddies do in bio. By the way, you're crushing that bit very, very severely right now. The point was, what I'm saying is, if you got a process, you don't mess with it. What you do is you clone it. You don't make it bigger, right? So if you're making 1,000 liters, tomorrow you don't make 10,000 because that's a bright idea. That's economic suicide in certain cases. However, you know, I'm hearing this, and I'm hearing continuous process and intensification. So, just to come back to it, I mean, is to get the stuff through the goalposts, is it still the process is sacrosanct? We don't want to mess with it, or you guys? Why don't we start from there? Is that a good question? I mean,
well, you brought up the question: is it smarter to scale out or scale up? Mm-hmm. Can we start there with that one?
Start with that one.
Yeah, okay. And the problem is there's no right or wrong answer. If you scale out, you're obviously not enjoying economies of scale. You're not benefiting from economies of scale. It takes one person to run this tank this size, and it takes one person to run a tank 10 times this size. It makes 10 times the amount of drug, right? That's economies of scale. Obviously, if you're scaling out this tank 10 times, then you're not enjoying the economies of scale, and you're not reducing your cost of goods as a result. However, you are duplicating the process, and by definition, you're not complicating your life from a process quality and CQA, right, quality attributes perspective. So there's less challenge there, right, and less risk of failure.
Right.
If you take the risk and scale up, if you're really good, you know what you're doing, and you might hit the goalposts. And it's always a question of, can we? You know, it's not a given. The question is, how good is your team at scaling up and controlling the critical quality attributes when they get to final scale? So those are the pushes and pulls, risks and benefits. There's no right or wrong answer.
So that really plays to the continuous, where it's this big, but it depends on how long I run it.
Exactly.
Is that right?
Exactly.
So why don't we talk about that?
Yeah. So one of the things that we've been doing is invest a lot in continuous manufacturing, particularly for therapeutic proteins. We feel that with continuous manufacturing, a couple of advantages. One is you can actually manufacture the required amount of protein with a much smaller scale. So for example, if you have a 200-liter bioreactor at say, a couple of grams per liter, having a bank of those 200-liter bioreactors running for a month, you can produce close to 5 kilograms of protein. So it's a much smaller scale. You do scale out like what Parrish talked about, but you also maintain the flexibilities. I make my 5 kilograms of drug in 1 month. I want to switch over to a different molecule. I can switch over to a different molecule. I haven't lost anything. And now my downstream is being operated for the entire month. So I'm essentially utilizing my capital that I've invested in the downstream process through the entire month. So I have a smaller facility. I have a better return on my invested capital. So overall, I can actually reduce my cost of goods. So now what companies like Pall and other companies are doing is trying to figure out how we can make the downstream continuous process actually work effectively. And that's essentially where a lot of the research is happening, a lot of the development is happening.
So let me ask you this. I mean, you brought up, obviously the proteins are the big challenge, right?
Yeah.
So what's going on there?
So the multiple challenges with continuous processing of proteins, right? First is not all the analytical technologies that are required to actually monitor and control doing feedback or feed-forward control. Not all the analytical technologies are currently available, right? So if you want to do inline measurement and then do a feed-forward or feedback control, we don't have technologies available today. So that's one of the challenges that we have. The second one is we're still trying to understand how you do deviation management, how you do perturbation management. If there's a process perturbation, what do you do? Do you throw away the whole batch? Do you throw away the whole month's worth of run? So how do you manage all those deviations? When everything is running steady state, life is good. When things start going awry, which usually does, that's when we need to understand how do you manage those kind of events. And then the third issue that we are also trying to wrestle with is what does QBD mean? How do you do scaled down validation? Right, when you have a continuous process, what's your design space? What does quality by design mean? And then how do you validate the scaled down process and demonstrate equivalence at a small scale level? So those are the kind of things that we are trying to work through right now.
And that's back to you. I mean,
that's why there's a lot of debate about should I go continuous or not. And we hear it around the globe again and again and again. People are asking us, asking others, should we, if we jump into biosimilars, should we go to continuous manufacturing or not? And so you have to step back and do a risk-benefit analysis. Again, there's no right or wrong answer. You have to look at what are the benefits and then what are the risks with regard to not scaling up, losing economies of scale. Number two, not having process automation. So you're basically producing blind continuously because you don't have real-time analytics telling you how well you're producing those critical quality attributes. And number three, in the upstream, for instance, people complain about how much media they consume in their continuous bioreactors. So there is no credible report today that we have seen and we have been watching documenting that continuous manufacturing reduces your cost of goods across the board. There is no definitive analysis available today that proves that definitively from any credible source. So there's a lot of debate today about whether or not being competitive in the biosimilar landscape from a cost perspective, manufacturing cost perspective, can be achieved by going continuous or not. Is there a possibility? Yes. All the breads have to land butter side up, as Manny is correctly pointing out. And if we can do that, if we can achieve that, then there's the possibility we can make step-fold changes in cost of goods by going continuous.
But I think the way we see this transitioning is a stepwise manner. We don't expect a continuous process all the way through, but we see islands of continuity. So you basically take a few unit operations and make those continuous, and then take a few other unit operations, make those continuous, but have some kind of a break in between so that you are giving yourself some time for some kind of deviation management or some kind of process feedback. So we see this not going all the way from the current batch process to a fully continuous process, but in a staged manner. So there is a lot of interest in looking at islands of continuity rather than a fully continuous process.
Well, what I like is when you allude to QBD, I mean, the bottom line is, and what we've always seen, is a firm's inability to leverage their institutional knowledge. We're talking about an innovator, for example. To me, and I've always been thoroughly convinced that a contract manufacturing organization even has a better opportunity to leverage institutional knowledge from that standpoint. What I'm trying to say is, if I come in and I say, I want to maintain interchangeability, okay, from a QBD standpoint, okay, talk to me. I mean, there's got to be a lot in institutional knowledge, process knowledge, development. The bottom line is, do I have to spend more? If I want to even consider, absolutely, you got to be interchangeable. Absolutely has to be a continuous process. Sounds to me like I'm saying I better have a hell of a lot more developmental work done. So let me add. So Parrish, from your standpoint, I mean, do I have a handle on it?
And I agree with Manny. You should walk before you run. Get the process working at a simple level, a batch or fed batch, per the original process, so that you've got a baseline and you've got your analytics working and you've proven that you've got biocomparability. Then do your cost of good analysis and efficiency analysis. From there, make selected changes to the bottlenecks in your process that you identify, and try innovations in silos on those bottlenecks and slowly remove those bottlenecks because they're going to move around. When you reduce this one, you're going to have another one show up over here. So you look at those and you reduce them to the point where you've brought in innovation to the extent, you've reduced the bottlenecks, you've reduced your cost of goods, and you're maintaining all along the way those critical quality attributes. And so the point here is not to try to boil the ocean at time zero. You know, you walk, and then you walk faster, and then you run if everything's going well.
So there has got to be obviously a COGS reduction here for manufacturing biosimilar. So what degree of reduction in cost of manufacturing? What are we looking at? You know, so,
from continuous?
Yeah.
I don't think
that
I would say any particular number. What I will say, we hear a lot less debate about going continuous downstream with regard to lowering costs. A lot less debate. A lot more folks agree that if they go continuous with downstream,
okay,
you know, continuous chromatography, et cetera, or connected systems, that you reduce buffer and media and resin consumption.
Absolutely.
Very consistent report back from the industry around the world. Upstream, a lot of debate. You know, you shrink the reactor tenfold, but now you have to add 2 more vessels to feed that vessel, to feed that reactor, and you have to add 2 more vessels to harvest from that reactor. So you shrank the reactor tenfold, but now you've added 4 sterile vessels to manage it under continuous manufacturing. Number 2, you've got a lot of media roaring through your bioreactor. So if you don't have the team on board, if you don't have an analytical team that can do spent media analysis to determine what the cells are consuming, only what they're consuming, then you're just going to blindly pump media through it at very high cost to go continuous. And that's usually where upstream perfusion economics flip over. They flip upside down because the cost of media perfusion is very high. And so we hear a lot of debate around the industry about the upstream and whether or not perfusion is economically viable. Downstream, a lot less debate.
I got you.
But I think, to me, there is a step between a fed batch reactor and a perfusion reactor is basically running multiple fed batches so that you're feeding the downstream constantly. So it doesn't have to be one or the other. You can basically have multiple smaller fed batch bioreactors constantly feeding the downstream portion, right? So there are different ways to skin this cat, and to me, I don't think necessarily you need to go directly to a perfusion process.
Yeah, I love talking about the process aspect, but I got to tell you, one of the things that we spent, it's not only does it really, what does it bring to the table, but believe it or not, one of the big, on the small molecule side, one of the problems was the regulatory dimension. So, you know, FDA has been, I won't say silent, but the bottom line is they kind of leave a rather wide door open on what we define as continuous. So what's the regulatory environment for I don't mean to keep harping on continuous, but you know, we're talking a year later, but anyway, where are we at with that? I mean, what's the regulatory sense of continuous?
At least, I mean, from my standpoint, we feel that the regulators are actually leading the way in this space. They're in fact encouraged, at least the US FDA has been very vocal about this. They believe that moving to a continuous process will actually help them in getting consistency of quality. And so they've been pushing very hard on the industry to adopt continuous technologies, albeit right now for small molecules, but even for biologics, they certainly are encouraging companies like us to come and talk to them, to help to educate them on the technologies that we have. We've done that already. And so they want to understand what new technologies are available and what these technologies can do in the area of bioprocessing from a continuous manufacturing perspective. They also understand it's not going to be a jump directly from batch to continuous. They understand that it's going to take place in phases, but they want to get ahead of it, and they want to help companies, whether biosimilar manufacturers or innovative drug manufacturers, to work with them to go down this path. So they've set up this emerging technologies team, the ETT, and the ETT has been very, very vocal about this. They actually want the sponsors to come in and start having conversations with them early on so they can work together to get the molecule approved. So they've been very, very open about it.
Well, you know, we're talking about biosimilars, and I just realized, I was just checking the time. What I want to get to, and I kind of slipped my mind, on the biosimilars. So the answer is, here we are a year later, there's been a lot of activity. So what's the regulatory landscape, Parrish? I mean, we got global launches, I mean, is there any difference between here, Asia?
It's
moving. Has it changed? Or?
It's moving, it's getting better, right? We have two We have 2 molecules approved by the FDA in the last year, and one is the more complex monoclonal antibody. More recently, Somma from Celltrion. So that's all good news, and that is obviously opening the door and providing clarity for companies around the world to approach the FDA, because there is a precedent for licensure of biosimilars in the U.S. by the FDA, and also obviously Europe is way ahead. On the continuous front, just to comment,
Yeah.
you know, there are 15 to 20 drugs on the market today produced continuously upstream, licensed by the FDA and EMA. Factor 8, it's a good example. Produced continuously. So continuous manufacturing has been around a long time, and the regulatory envelope around that has been established for commercial drugs for a long time. So that's not new space. The question here is whether or not it's appropriate for biosimilars in being cost competitive and productive in this new landscape.
So there's a couple of questions I wanted to get back to ask again, and that is, so obviously in an environment, again, where science and commerce meet, this is what we're talking about here. I mean, what advice, there's got to be some companies out there, what advice do you say, guys thinking about going into biosimilars, what advice do you give them? Don't. So help me out there. Who wants to tackle that one, Manny? You want to do that? Convince me.
I think it's an exciting space to be in. I think there is a lot of unmet demand, not just in the US, but around the world. I think there was a study done by one of the companies, and they said less than 28% of the eligible patient population can actually afford some of these therapeutic biomolecules today in the US. So certainly there's a lot of demand out there for these molecules that cannot be met by with today's costs. Right? So there's an opportunity, but I think from a biosimilar standpoint, developing a molecule at 70% the current innovator's market price point, I don't think is going to make a huge dent in the marketplace. If you really want to make a dent in the marketplace, you need to come down to much, much lower costs in terms of in-market costs. And that's where the pressures on the cost of goods is going to come really, really heavy. Right? So, for example, I think in Norway, they're talking about some of these molecules at 70% discounts of list prices, so significant discounts of list prices for the country to be able to afford those molecules and have them a part of their national health system. So the costs have to come down, but I believe that this is a space where the demand is there, and with some of the new technologies, we can certainly bring the cost down. And it's just up to companies to start investing and jumping into this space. Yeah. It is possible.
From our side, I can tell you, there's some of the small molecule guys, it gets to the point where if they're not making it, they walk away. I mean, yeah. I hate like hell to see that go down in another industry. But, what do you think, Parrish?
Well, I agree in general. It's a very exciting space. Yeah. We're going to see it drive a tremendous amount of innovation over the next 5 to 10 years. That in and itself is going to be very interesting. The real question is there a business case for any company getting into a very competitive market? And so you have to ask the question from a number of perspectives. Do we have the cash to fund it and to build the plant and to hire the people to do it? Do we have the analytical horsepower in-house? Do we know enough about the molecule? What are the cost objectives? What are the cost of goods and pricing, objectives that we have to hit, and put that all together and make a complete business case. As technologists, as technology providers, we can help solve the problems about scalability, product quality, the right technology, doing assessments of batch, fed batch, continuous, which should you do from a process economic perspective. But before all of that, the question is, what's your target to be competitive in the market that you're going to go into with that molecule? What's the business case?
So it would be legitimate to say, and this, again, we've talked to a lot of folks on the panel, and what we've starting to see is the contract sector seems to be actually moving into the forefront on pushing ahead. And you see they're moving into continuous more so than the innovators are. And again, not necessarily just as an example. So they have obviously they got a vision for the next 5 years. So what do you guys think? Let's fast forward 10 or 15 years here, and we'll take it in turns. So where are we going to be, Manny? I mean, here we are a year later, and we were able to fill up 43 minutes about it. So now I figure 10 years I give you, so, you know, so where's that at? What's the Jules Verne version of that thing? What is that?
So just to go back to your innovator companies not going down this path in terms of continuous or some of the new advances. So the innovator companies have an installed base already. So there's a reason for them not to go down this path. But we see a lot of interest in the smaller companies, smaller mid-sized companies, including some of the CMOs, that see that there's an opportunity for them to create a differentiated product, differentiated manufacturing process that gives them an advantage in the marketplace. So we see that going not necessarily with the large innovators, but with some of the mid-tier and the smaller innovator companies, and also biosimilar manufacturers. So we see them moving in the new direction. To answer your question in terms of where we see this, I think, you know, to me, 10 years from now, I would expect that there will be a few manufacturers around the world who are able to produce a therapeutic antibody at $1 per day in terms of a patient cost, $1 a day of patient cost.
I like that prediction. Probably every insurance company in the world would love that prediction. Yeah, that's for sure.
They will get there.
I got you. I mean, yeah. When you consider what penicillin cost originally, all right.
Yeah.
It was
It used to be 10 mg per liter. Now it's 70 grams per liter in the fermentation.
And that was stuff that they would recover, included, right? So we won't get into that. But, so Parrish, where do you see it? I mean, to me, obviously, here we are.
I think we're going to see a broad spectrum of a mix. You're going to have Celltrions and Samsungs that are knocking out 15,000 liter plants, all stainless, and they're buying market share by lowering their price 70%, just as we've seen in Scandinavia for Remsima. And then there are going to be local markets where there is national protection. That is, the national government is going to say, you can't import your biosimilar into our country unless you produce in our country and hire our people, and we're going to guarantee your market because we're going to buy it for the populace. So there's going to be a spread of global players and local players. And therefore, we're going to see a spread in capacities and size of facilities. And I think that there is always room for innovation, as long as we maintain product quality, to help us lower costs in general. So we can't predict the future, but I think there's enough variability there. It's going to be very exciting, very interesting, and should be tremendous next 5-10 years.
That's something to think about. Yeah, I mean, the other thing that always comes to mind, and invariably we had a fellow in here earlier today that talked about drug shortage, and it seems to me that there's another way to address it. Obviously, it's a great opportunity. I don't think the time that we have left is going to allow us to get into that. We had a few minutes. Any questions from the audience here? We'll entertain that. Other than that, we're going to gracefully bow out and finish up. Any questions? Don't all jump up at once, by the way. You know, we don't bite, honestly. So, no question? No? Okay. Well, thank you very much, gentlemen. Manny, Parrish, appreciate it.
Thank you very much.
Pharmaceutical Processing, in conjunction with INTERPHEX 2016, presents INTERPHEX Live.